Okay, well thank you Rick for giving me the opportunity to talk today. So just to explain, I'm actually an endocrinology registrar, but I did my PhD in this area a couple of years ago, and I have ongoing research interests in this area. And really all of this work started off with a remarkable lady who I met back in 2010. And as you can see, she has a very unusual problem in that she has massive overgrowth of her legs, but a very lean and normal sized upper body. And she was in fact born with this condition. So you can see she had very large legs as a baby, and they have progressively grown throughout her life. Now when we look in detail at her legs, what we see is in actual fact she has massive expansion of fat in her legs. And she also has an area of fatty overgrowth in her abdomen as well, which is very well demarcated in the midline. So in view of all of these features, we really formed the impression that this lady probably had a Mosaic overgrowth disorder. So that is a disorder which affects overgrowth in one part of the body but not in other parts. And probably the commonest example of this, or the one people know most about, is Joseph Merrick, who was known as the Elephant Man in the less kind Victorian era, who was believed to have suffered from one of these disorders called Proteus syndrome. So in order to investigate our lady, we took a biopsy from her arm, which we considered to be unaffected tissue. And we also had a sample of tissue from her leg. You can see that it's been amputated. So we considered that to be affected tissue. And we performed a very powerful genetic analysis on it called whole exome sequencing. And we asked ourselves the question, what genetic variants are present in the leg that are not present in the arm? And actually in doing that, we fairly quickly came to what we believed to be the answer. And that was a genetic change in the gene PIK3CA. And we found that genetic change only in her legs, and also we found it in varying amounts in different tissues in her body, which again suggested that this was a mosaic genetic change. Now, the reason why we thought that was probably the answer was because it's a gene we were very familiar with as endocrinologists, and it encodes a protein called PI3 kinase, which is critical. It's in all of our cells, and it's involved in a lot of different processes, including cell growth, and also sugar handling, fat synthesis, and other processes. It's also a gene that's very well known in the cancer world as well. And this is an extract from Cosmic, which is the catalogue of somatic mutations in cancer developed by the Sanger Institute. And this particular mutation that this lady had is actually massively overrepresented in cancers as well. So because of that knowledge, we knew very quickly that this particular genetic change causes the protein to, if you like, gain function. So the protein becomes too efficient. And I tell my patients it's a bit like a traffic light being stuck on green all the time. So as a consequence, we were seeing increased cellular growth, obviously in the cells that are affected, which is responsible for her phenotype. Now, we were quite lucky to publish this, but literally simultaneously, in about a month of us publishing this, all these other reports came out from across the world, describing a whole range of different patients with these particular mutations. And now we really recognise that in fact there's a spectrum of conditions caused by these genetic changes. So at the milder end of the spectrum, we see patients with just an enlarged finger or thumb or an enlarged limb. We see patients with blood vessel problems and massive overgrowth of blood vessels, bleeding, and we also see patients with brain overgrowth. And that may be very mild, with a mild learning disability, or it may be very severe and the patient has intractable epilepsy, developmental delay, autism and brain herniation. Now for these patients, they weren't really recognised before, and of course there aren't really any treatments for them. For the patients with overgrowth, they have to go to a surgeon every couple of years and often have radical surgery to chop off bits of tissue. Because no one was really able to connect them all together, no one really knew that they were caused by the same thing. So following this work, we really set up an observational research study called the Segmental Overgrowth Study to help investigate more in this area. And we got this approved in 2013. And as you can see, the uptake of the study has been huge. We've recruited 270 subjects in just under 3 years. And in doing that, we've been able to make 90 new genetic diagnoses. We're part of something called the Musketeers Memorandum, which I think is really useful to know about in rare disease research. And this is like an overarching agreement across the UK where you can use lots of different hospitals with just one agreement to do recruiting for you, consenting, blood tests, and so forth. So that's really helped our recruitment. The other thing that's really helped us is social media, so Facebook, Twitter, the website. We get 50% of our referrals now through this medium from patients themselves. So really the aims of our study are threefold. First, we want to make a genetic diagnosis for our patients. The second thing we want to do is to be able to study the natural history so we can understand more about these conditions. But the third thing we're very interested in is cohort building for clinical trials. And the reason for that is because this is a gain of function mutation that's switched on, you have the ability to potentially go in with a drug to switch it off, so switch that traffic light back to red. And it so happens through the links to cancer and also a little bit of serendipity that there are actually some drugs available which can do this. So first there are drugs in cancer phase trials, phase 2-3, which will specifically go in and inhibit PIK3CA, which is where we have the problem. But obviously this isn't available to us yet, this is still in phase development. But there are other drugs, and one in particular we've heard a lot about today is Sirolimus, which is an mTOR inhibitor, which can act downstream from this area. Now obviously, AKT, PI3 kinase, these are all proteins which are very important in insulin action, and they have multiple different roles in cells. So they may affect growth, they may affect the ability of a cell to suck up sugar, for example. So you can see how Sirolimus can help in both of these types of conditions. So we decided to look at Sirolimus in more detail just for the reason that it was available. We knew there was long-term safety data for it, and obviously it was off patent as well. So I started off, and I did some of this work during my PhD, looking at cells from some of our patients. And what we were able to see is that Sirolimus very effectively switched off growth signals in the cells from patients. And not only did it switch them off, it did this at remarkably low concentrations as well. So this was really exciting to us, because if we were going to treat our patients, we wanted to be sure we could treat them with a dose that was low enough that would avoid them having lots of horrible side effects as well. So following this work, we went back to our original patient, and we saw how she'd been getting on, and she was really getting into a bit of a sticky wicket. She was putting on more and more weight in her legs. She was unable to rehabilitate. She was outgrowing the prosthesis she had in her left leg, and she was absolutely terrified that she was going to become bedbound. So she really wanted us to do something to try and help, and we discussed with her the possibility of Sirolimus treatment, and she was understandably very keen to give this a try. So we prescribed this as an off-license treatment, and as you can see, the response was really quite dramatic. We started her in a tiny dose, so this is half the dose that Scarlett is having, given the size of this lady. And we used this dose partly because it was an unknown quantity, but also from our cellular studies suggesting only a little amount of the drug would be needed. And you can see that over 3 years she's had a sustained response to Sirolimus and has lost tissue from her legs. And if you don't believe the graph, you just need to look at the DEXA scan outline, which is how we measure tissue changes in her. And you can see that before she started Sirolimus, she didn't fit inside the box, whereas now, after taking Sirolimus for 3 years, she does actually now fit right inside the box, suggesting she genuinely has shrunk. Now, the problem with this, obviously, if we want to be self-critical, we'd say, well, how do you know this isn't just a massive placebo effect? And you give a patient who is desperate for a treatment, a tablet, and say, this is going to work, and they don't go away on a massive crash diet. So we were actually very keen to look for any collateral evidence that it was actually working. And that came in the form of this skin lesion at the base of this lady's spine. And we knew that this was part of her disease process because we could detect the genetic change in that skin lesion. And what we saw following treatment with Sirolimus was that that skin lesion virtually vanished. So again, that gave us added, if you like, evidence that the treatment was working. So we really felt from that point, the natural progression was, of course, to move towards a clinical trial with Sirolimus. And the actual trial design that we have decided upon is an open-label, uncontrolled, non-randomised pilot trial. And to some degree, it does pain me to say that, because obviously you've heard a lot about RCTs today. And as a physician, the gold standard is a double-blinded, so the physician and the patient doesn't know which treatment, randomised to different arms, controlled with a placebo or a therapy, another therapy which can be used in that condition. And that's the gold standard. And really that's the evidence that we need to go to people like NICE and say, we think this drug works, and we think this drug should be available for other patients. But obviously we were doing this sort of less robust trial form, which may not even give us an answer. So obviously I will go through our reasons for deciding on that trial design, but I think that might be a common thing that people in this arena actually encounter when they're repurposing drugs for rare diseases. Now, our trial design, we're including 3 to 65-year-olds. We have 10 patients in the UK, and we're doing just a baseline scan for 6 months and then treating patients for 6 months. So it's very, very simple. It took this long to get this trial up and running, 17 months. So in September 2014, we decided with our American collaborators we'd do this, so we did high fives and all those sorts of things. We set up the trial design, lots of conference calls, very enthusiastic. We ensured we had funding, we had all our collaborators in place, and really within 4 months we'd done our entire set of documents for the trial, and we submitted them to our clinical trials unit. Now, what is a clinical trials unit? Across the UK, really any clinical trial you do within a hospital, the sponsor will want you often to interface with one of these bodies because they're endorsed by the MHRA. And that review and insurance and all sorts of other things took a further 8 months. It was like walking through sludge. It didn't seem to matter what you did. It just took so long. Eventually, we got to submission to the regulators, so the Research Ethics Council and MHRA, and that took about 3 months to do all of the sort of responses and so forth. And then finally, we had the final hurdle, which was all of the internal approvals of bits of paper going to probably in excess of 20 people within our trust to ensure that it was safe for us to start the trial. And I'm very pleased to say that 2 weeks ago we enrolled our first patient, and we had our second just a week ago. So we're finally there. But the question is, why has it taken so long? And it really, I think this comes down to the level of regulation in clinical trials with a medicinal product, so known as a CTIM. And the reason for that is that we're bound by the EU Clinical Trials Directive of 2001, and really it encompasses all types of trials of medicinal products. So you would have the same kind of regulation even for a phase 1 trial with a completely new drug in comparison to something like this with Sirolimus, which obviously has been used for 14 years. So what other challenges did we come across in this trial design? So firstly, a rare disease, and this is a problem everyone's going to encounter, and I think it was Costello, the consultant, who alluded to this earlier, that we have often a very limited overview of the natural history of these conditions, just simply because they are so rare and there aren't enough patients to be able to measure things. And that was really the reason why we had to move from an RCT to a pilot trial, because we just simply did not have the data to be able to statistically power a more robust trial. We also came across the problem of a creep in off-licence prescription, and I think this is a really difficult area in our field as well, repurposing in rare diseases. There are situations where off-licence treatment is absolutely the right thing to do, and I think with Scarlet we had a very, very good example of that. But clearly it's very difficult when you have someone who has a chronic, stable condition. They find it very debilitating. They are desperate for a treatment, but obviously we don't know whether the treatment actually works or not, and that's why we need to do the trial in the first place. The last thing we want is for treatments to become entrenched in clinical practice without knowing whether or not they're actually doing any good or just causing harm. So it's very, very tricky, and in our case we found that actually it was from the American side that a lot of patients in the US were being treated off licence with Sirolimus, and because of that our American colleagues said to us, look, the only way that we can recruit enough patients into the trial will be if we do an open label trial and an uncontrolled trial. There's absolutely no way our patients will accept a placebo-controlled trial. So now obviously it's a very delicate balancing act. It's not the most robust trial, but equally you want to do something that's acceptable to patients. So I think that's where PPI is so important in these things, to try and strike that correct balance. Now obviously with it being rare, particularly when we started off this trial, we only had 10 patients in the UK, and our sort of magic number was 30. So naturally you have to collaborate, and for us we were very lucky. We already had existing working relationships with a group in the US and French, which expanded our potential patient pool to 100. However, what we didn't anticipate was the problems of having patients scattered throughout the UK, and of course your patients are in Wales, they're in Plymouth, they're in Manchester, and we're in Cambridge. And of course you want to ensure that you're keeping your patients safe throughout the trial and doing all the correct safety manoeuvres, but equally you don't want your patient travelling for 5-hour round trips every week as well. So I think now I realise quite naively we wrote into our protocol that we would just do blood tests for safety local to the participants, and we would do that with local hospitals, local GP practices, and we didn't really think anything of it. And obviously coming through the other side, we've now realised our error. So the first issue we've had are NHS trusts telling us they have no capacity to do blood tests. So even if it's one patient and it's 3 blood tests, we've been told no, even if we say we're happy to pay. GPs, we had to get CCG approval for this. It took 2 months, and when it came through, we found out it was England only, and we'd have to repeat the same for Wales or Scotland or Northern Ireland. So really, I think eventually we've actually had to move into sort of using the private sector to do this, because obviously the cost-benefit analysis of having a private practice do it for £25 versus the patient travelling back and expenses, it's more cost-effective. But it's worth being aware that that is the environment in the NHS at the moment. So I think really the things I've learned, we've all been talking about this, collaboration is so important. I think I've really learned minimise your blood tests and study interventions. Don't get too academic or interested about this, just do what you need to do to be able to prove what you're doing. Building flexibility in your protocol about private sector involvement, because you may well need that and it may well save you costs down the line. And then one thing I've wondered about just doing this work, whether it would be worth looking into collectively, whether a standardised agreement for blood tests in rare disease and repurposing in the UK, particularly with the links with the NHS, might be a sort of feasible thing to do and something potentially Findacure could help with. Now the second challenge we face is of course funding and costs, and this is a non-exhaustive list of all of the things you'll think about in your budget for a clinical trial. And this is actually a snapshot from our clinical trials unit of the amount of money they quoted us for this 6-month-long trial, which you can see is of the region of 80,000. And in fact, the entire trial was quoted to be over £150,000. Now, we had actually just cobbled together a little bit of money from the backs of research grants and things that we had, and we had little pots of money here and there, but we certainly didn't have 150,000. So we were pretty shocked when we saw this. And again, I think this really emphasises how important the work Findacure are doing in helping to sort of meet that gap. But obviously we didn't know about Findacure, so we really went into the business of trying to cut down this bill. So the way that we did that was, first of all, we contacted Pfizer, the manufacturer of Sirolimus, and we knew that it was off patent, and we knew that there was a good chance that they wouldn't say, yes, of course, we'll just bankroll your entire trial, but we thought they may be able to help. And they did. They provided us with drug free of charge for our trial, and that really helped our French collaborators to come into the trial. So it's been really beneficial to us. The database. Now, I had a particular bugbear with database. We were quoted £30,000 for a database for our trial. And again, this is a regulatory issue. You have to have a specific database with specific audit trail, security, and so forth. And I'd actually worked out with our 10 patients and 8 data points, we were going to have 80 data points and a trial and a database costing £30,000. So after a lot of nagging of the clinical trials unit, I was eventually able to wheedle out of it. But in actual fact, the MHRA are pretty happy with an Excel workbook, which of course is free. So immediately shared 30,000 of the bill with that. And I think that's very worth knowing about if you've got a trial with a small number of patients. Now the other issue we came across is obviously with 2 other partners we were working with in France and the US is a multi-centre trial, and obviously we wanted to be the sort of main centre in Cambridge and to have the French and US as study sites, and immediately were blocked by the costs of insurance, which were said to go into possibly tens of thousands for this type of trial. For our insurance in Cambridge, obviously we're covered by the university, but you have to pay an extra premium sometimes if you want to expand out. So for that reason, we actually set this up as 3 independent but identical trials, all following the same protocol, which obviously cut out that cost. Now this isn't without issues. It's very tricky when you have local involvement, you have recs and all sorts of people who are adjusting and chipping away at your protocol, but certainly it did save a lot of money. Now, drop shipping was another problem we came across. So we thought, right, well, we have the agreement with Pfizer, we'll get all the drug in Cambridge and we'll ship it out to the US and France. And we got a quote for doing that. And again, lots of regulation, lots of QC processes, and we were quoted in the region of £30,000 to £40,000 just for that alone. So again, that was something we didn't really want to pay for, and in actual fact, to circumvent that, we went back to Pfizer, and we all applied independently, and Pfizer were really flexible about that, and in actual fact, we solved the problem. So after all of that sort of battering down, we managed to get the trial in the region of 60,000, 70,000, which is much more palatable. I would add that doesn't include obviously time of investigators and things, but we're often very happy to do that, just as part of our sort of normal working day, really. So I think really what I've learned is that always speak with pharmaceutical partners, even if you think it's off patent and they're not going to be interested. Generally, we've had a really, really good response. Haggle with the clinical trials units. These bills are not set in stone, and I think you can really try and push the numbers down. And then obviously, again, just simplify your trial where possible. So now, obviously, I've mentioned a lot about regulation, and I think it's widely recognised that the Clinical Trials Directive of 2001 had some problems. And in actual fact, some of the figures suggest that there's been a 25% reduction in the number of trials, and also a doubling in the cost of trials since this directive came in. But the really good news is that they've recognised this. It's taken about 13 to 14 years to do so, and they have revised this legislation, and it's due to come online in 2018, but they've brought up the concept of a low intervention trial, which is a trial with minimal additional risk compared to standard care. And I think there's a very good chance that many of the trials we'll be doing here with repurposing will fall into this category. They're going to have much less stringent rules, less costs around monitoring, trial master file contents, and I think it's going to massively cut down the costs and also simplify for what we're doing. So if you've got your trial planned for Q 2017, for Q4 2017, I would suggest that you keep an eye on this, because actually you can save a lot of money when this comes into force. So it's really good news on the horizon. So finally, just to conclude, I've talked to you a little bit about conditions caused by mosaic mutations in the PI3 kinase signalling pathway, and shown that they're likely to be potentially treatable with drugs such as mTOR inhibitors and others. And that we finally have a trial repurposing Sirolimus. And obviously, this is notwithstanding some challenges, but what I would say is I think if you're motivated in this area and you have enough tenacity and perseverance, you will eventually get there. And just to acknowledge all of the people I work with, collaborators, our physicians and their families, and any precedents.